Hello and welcome to the online symposium on the state of COVID-19 in Bahrain. My name is Professor Stephen Atkin and I am the head of postgraduate studies and research here at the University of Bahrain. COVID-19 disease has led to unprecedented times with a complete change on how everyone in the world has conducted their day-to-day business. And the societal effects are still having repercussions now. Despite a relative return to normality, this could change at any time with new SARS-CoV-2 variants. The global impact that we all felt has been at several levels. There's been a personal impact that has reflected through increased mobility of the disease, the increased mortality of the disease, and the pain that almost every individual has felt in one form or another. There has been societal impact through social distancing, and the normal fabric of society has been disrupted. There has been an economic impact, both on individuals through their employment being compromised and total global slowdown. There has been a need to combat this infection by increased understanding of its infectivity and mode of infection, disease progression, the impact on vulnerable populations and who those populations were and are, treatment of the acute infection in mild, moderate and severe disease scenarios, and prevention of the disease through vaccination and public health measures. In this environment, the Kingdom of Bahrain has been at the forefront of this pandemic through leadership of people such as Professor Manaf and the inspired work he has led. This work has been recognised on the global stage and has genuinely contributed to the fight against COVID-19 disease that has been recognised and acknowledged by the World Health Organization. This mini symposium today is to highlight just some of the work that has been performed and is ongoing from the Kingdom of Bahrain, and is a celebration of the research excellence that has resulted. We have 4 presentations today, followed by a question and answer session at the end of the symposium. Please send your questions through to me as you think of them, and I will present them at the end of the 4th presentation to the respective speakers. I would now like to invite Mr. Saad Mullah to present his presentation on the COVID-19 viral load not associated with disease severity, findings from a retrospective cohort study. Saad, the floor is yours.
Hello. So I'll be covering our findings from a retrospective cohort study that we conducted to investigate whether we can predict COVID-19 disease severity from a simple marker such as the CT values of RT-PCR diagnostic tests. So this study was actually born out of a curiosity that comes with having access to large amounts of data, thanks to the efforts of the National COVID-19 Task Force. And so it wasn't a primary question, but rather a secondary one that was born from that curiosity. And it was a very valid one as well, and one that has been asked and questioned repeatedly over the years with a lot of different infectious diseases. And COVID-19, of course, was no exception, specifically with the global context that came with it. And not only did it make this question a plausibility, answering this question a plausibility, but also even more valid and more important than previously. So some important background information. The gold standard for COVID-19 diagnosis is the reverse transcriptase polymerase chain reaction. As a lot of you are accustomed, it's done through a nasopharyngeal swab, ideally. And I have an illustration on the right as well to demonstrate how it works. But the basic principle here that we base this question on is the cycle threshold value, or the CT value. And this is a number that it comes with every RT-PCR test. And it refers to the number of replication cycles that are needed to detect viral nucleic material. And so every patient will have a certain amount of what we call a viral load within them. And the assumption is that if a nasopharyngeal swab is conducted properly, then the amount of nucleic material in the sample is going to be proportional to the amount in the patient or in the human body. And so the more viral material there is in the sample, the less cycles of replication will be needed in order to reach a threshold that you can fairly and accurately detect COVID-19 or SARS-CoV-2 specifically in that sample. And so the concept here is that the smaller the number of cycles that are required to reach that detection threshold, the higher the viral load. And so that's the concept or the principle that we based our question on. And there has been real-life data that has attempted to correlate viral load with disease severity, and RT-PCR CT values is only a surrogate. I have to mention that it's only a surrogate to viral load because we can't specifically, there's a lot of factors that might come into why CT values may differ. And one of them is just the way that the sample is collected. Just something as simple as that can affect it. And so it's a surrogate marker that's meant to indicate viral load in a specific patient. And there has been some studies on that, but the results have been disputed. And so the aim of the study was to be able to use COVID-19 RT-PCR CT values as a simple marker of disease severity that would allow us to easily and proactively identify and triage high-risk cases. And specifically, I think COVID-19, the pandemic, has shown us the importance of, or the large role that health resources in general play, and health equity as well. And so it's very important, not just with COVID-19, but with other infectious diseases and with future infectious epidemics and pandemics. It's very important to be able to have a simple tool that we can use to identify whether someone is at high risk for severe disease or whether they're not at high risk. And that would then determine the amount of resources that we're going to dedicate to each person. And so this study then sought to investigate for clinical correlation between CT values and disease severity, specifically on prevalence. So to do so, we conducted a retrospective cohort study on randomly selected COVID cases who were admitted to Ministry of Health facilities from May to July 2020. And the collected data included patient demographics, as well as CT values on diagnosis. So that was the main variable. And then we also collected a lot of data on comorbidities, like sickle cell, G6PD, diabetes, obesity, just so that we can be able to exclude some confounding factors or investigate for any confounding factors. And then we also took note of the medical regimens that the patients were on if they were admitted, and of course the respiratory status on admission, which was the second important or main variable, and in this case the dependent variable. And so the three main different types of status was room air, oxygen support, and non-invasive ventilation / high flow nasal cannulation.
So in terms of the results, we had a total of 1,057 cases included in our cohort. The mean age was 46.26 with a slight male majority. The symptomatic rate was actually 67%, which is quite low. And the reason for that is most likely the heterogeneity that was in our cases. So some of them would present with severe disease just on presentation, whereas others would simply be close contacts of positive cases, and that would be the reason why they tested, so they would be asymptomatic, whereas others, for example, would simply be doing it for travel purposes. And so it's a reflection of a good screening program when you have a good or you have a low symptomatic rate, because you're covering as much of the population as possible. Our comorbidities, we found hypertension to be the most common, followed by diabetes and G6PD, and then after that, there was cardiovascular disease, asthma, and chronic kidney disease. So in terms of the CT values, room air, patients admitted on room air had a CT value of around 24.9, which wasn't actually much different than those admitted on supplemental O2 or those admitted on non-invasive ventilation / high flow nasal cannulation. And surprisingly, or at least different than the hypothesis that we initially had, the CT values were actually higher in those who presented with a larger need for oxygenation, which in this case was a marker for disease severity, or what we use as an indicator for disease severity. But interestingly, when we refer to this categorically and we try to create categories of patients in the highest viral load, which was a CT value between 10 and 20, and patients on a medium viral load, which was a CT value between 20 and 30, and then comparing that to those with the lowest viral load, which was a CT value more than 30, we found that the largest proportion of those with the highest viral load were those who required non-invasive ventilation. And then that was followed by those who were on room air and supplemental O2. And then the majority of those on room air and supplemental O2 actually had a medium viral load. And then interestingly enough, those with the lowest viral load, the majority of those who required non-invasive ventilation were actually in the lowest viral load category. And so you'll find that then those who presented with the most severe disease or the highest requirement for O2 on presentation or on admission were actually mostly formed from people on both ends of the spectrum, both highest viral load and lowest viral load. And we'll get to that later on as well, as reasons for that in the discussion. But that was an interesting finding.
And so you can see here that there is not much of a difference, or no significant difference between those who were on room air and then those who required oxygen support, regardless of the type of oxygen support when it comes to the CT value. And then in this case, what I was discussing later on in terms of the severity on admission compared to categorical CT values, those who were admitted on non-invasive ventilation had the largest proportion of those with the highest viral load, as well as the largest proportion of those with the lowest viral load, whereas those who were admitted on oxygen support or on room air mostly had patients in the middle CT value range, which we categorize as a medium viral load. And we conducted a logistic regression, and we found that after adjusting for confounders using the multivariate regression model, CT value showed no statistical significance for an association with the requirement for oxygenation on admission, and the odds ratio was 1.046, and the p-value is not significant. Interestingly enough, some of the findings that were obviously not the primary findings here, but what was significant with oxygen requirement on admission in terms of the confounding factors was age, as well as Bahraini status, whether they were Bahraini or not Bahraini, and I'll discuss this as well later on, as well as CKD and having specific symptoms such as a fever and CKD in terms of comorbidities. So despite a hypothetical plausibility that the role of or that viral load would have a role in the severity of respiratory infection, this has also been widely contested. In fact, in some studies, there have been differences within the same family of viruses. So, for example, the viral load of influenza A, H3, and 2 was found not to be associated with hospitalization, whereas there was data on the high viral loads of influenza B being associated with hospitalization. And in terms of COVID-19, there's actually been a systematic review on this, and it included 14 studies, and 8 out of those 14 studies found that there has been a correlation between CT value and viral load, and that there has been the significant association between disease severity and the viral load. However, there were 6 studies that found no significant association, and interestingly, our study would be the largest cohort. In fact, it would be 6 times the size of the largest one reported in that systematic review to report no association. But understanding the context here of our study is very important to understand any unaccounted for bias. So like I mentioned before, the patients that were included were very heterogeneous. We included patients who were tested in a cohort because they were either contacts of a positive case, so they were not necessarily symptomatic, We also included people who were symptomatic and they presented with symptoms, and even people from the emergency department, so who presented with severe symptoms, and so there was a lot of heterogeneity in that sense. And this is very important because of a concept which is time since onset of symptoms. And that's a very important concept and a limitation in one sense of the study, that we did not account for time since onset of symptoms. And the idea here is that viral shedding may have been taking place for a longer period of time in a specific patient, which would lead to the patient CT value being higher on admission compared to patients who only recently acquired the infection. But this patient may be someone who was staying at home and not presenting early on during disease onset, and so naturally they would present with more severe disease and a lower viral load, simply because they let the virus incubate for a longer period of time, and eventually the viral load would decrease, but their symptoms have been progressing. And so in another case, for instance, someone may have been just recently infected, or maybe a positive case, or a positive contact, or sorry, a contact of a positive case, and they would present very early on, and they would have a high viral load because they were just infected with SARS-CoV-2, and they would have a higher viral load and no disease on presentation. When in reality, had it been that they were not aware of it and they stayed at home longer or presented much later, they may have presented with more severe disease. And so that might be one explanation for why we had those who were those who required non-invasive ventilation, or those who were on the end of the spectrum. That may explain why they had CT values on both ends of the spectrum. Basically. And this is something I mentioned earlier, that nationality was an important factor. And so during this time the study was conducted, a lot of the non-Bahrainis formed migrant workers. And the migrant workers formed a very different health community in a sense, because they had lower health literacy and they would present much later. And they would also, they would live in a closer household, and they would be exposed, at least theoretically, to higher viral loads. And so all of these may have been factors that would impact the conclusion that we reached only from a biologic standpoint. So there's two aspects of this, and this is a conclusion that I'm trying to get to. There's two aspects to this question that we're asking, and one of them is, biologically, is there an association between viral load and disease severity? Is it the case that those who are infected or exposed to more virus, is it the case that they will be have more severe disease simply because they have more viral particles within them? And that's a biologic question that our study does not particularly answer. Our study answers instead the clinical question, which is CT values, or the way that we diagnose, or the moment that we diagnose infectious disease on presentation. Can we say, or can we use CT values, which are a surrogate of viral load? Can we use them to clinically indicate whether someone is at high risk for severe disease or not? And the answer to this is most likely not. And the reason for that is the heterogeneous nature of your patient cohort that I was mentioning. And so reliably, you can't use a simple marker such as CT values to not just indicate the viral load of the patient, but also indicate whether they're at high risk for disease, for severe disease or not. And so that's the conclusion that we reached. We did not find a significant correlation, and it did not seem to be a viable metric as a simple, unprocessed, and independent indicator of disease severity. However, whether it can be used as one part of a multivariable model, that's something that needs to be studied. And whether there is an association biologically or physiologically, that's also something that needs to have different study design to confidently reach a conclusion. And you can read more about our report in BMC Infectious Diseases. Thank you.
Saad, thank you very much indeed for your presentation, clear, concise, and also thought-provoking as well. And I'm sure that is going to lead to quite a few questions at the end of the session. So what I'd like to do now, it's my great pleasure to ask Professor Manav Al-Khatani to speak. As I say, Professor Khatani has been particularly, it has led Bahrain's charges that were against COVID-19 has been incredibly effective, and would like now to talk about randomized controlled trial of convalescent plasma therapy against standard therapy in patients with severe COVID-19 disease. Professor Manaf, the floor is yours.
All right, so good afternoon, everyone. Thank you very much for this invitation. I think this is our second year in a row that we talk about our COVID-19 research efforts. So I'd like to say a big thank to the all COVID-19 research team. I know Saad is among of this team, Abdullah, and of course, Professor Atkins. You've been very supportive during the last 1.5 years. So if you go to the next slide. So I'm going to spend just maybe 5 minutes before I go into my plasma therapy presentation. Who would like to say a few words? If you go to the next slide. So clearly, things I have learned personally when it comes for COVID-19 is data, interpretation of the data, and collecting the data and making them into an action policy are the only way you can find COVID-19. This comes to my brain vividly back in 2020 February, when we had the first case on February 26th, the first case of COVID-19 that landed here in Bahrain. At that time, there was really no information that tackles treatment, how do you contain this virus, the nature of this virus, transmission of this virus. And there has been really lacking of any data. And when you have a pandemic, this is going to be the chaos. This is going to be that if you're going to really wait for other people to present their data and their policy, you're really too late to fight this pandemic. And I have to say, Bahrain have never been engaged in data collection and even in publication heavily. And I'm very glad, and again, that's a big thank to the RCSI Bahrain, that in the last 2.5 years, we really have produced and published 31 publication, 29 publication plus 2 recommendations, and there has been a humongous international collaboration with Stanford University, there has been with the University of Columbia, and with different countries, and of course with other foreign embassies here in the Kingdom of Bahrain. So 29 publications, and all of this 29 has been really tackled all the aspect of COVID-19 over the last 2 years. So we have handled the epidemiology of the virus, the dynamic of the viral shedding, to the diagnosis. I remember when we talked about saliva testing versus nasal swab versus nasopharyngeal, when we tackle also about some test, a rapid test, antigen test. We also tackled the clinical, so we had over 16 publications on clinical and treatments. And again, we have done, I think, the first 2 randomized control trials in the Kingdom of Bahrain, the plasma and the favipiravir versus hydroxychloroquine. Again, feel very proud that we have established the infrastructure of having a very complex trials such as RCT. We also have 3 publications on vaccines. And again, Bahrain has been the second country in the region when been involved after the UAE in one of the biggest trial when it comes for the inactivated vaccine. We also have tackled the differences between the homologous and the heterologous vaccination. And finally, we contributed with the WHO in writing 2 recommendations about the maritime and things related to the COVID-19. Next slide. I have to say there are some preprints and there are some also ongoing projects. So we have, for example, currently 4 submitted papers. So one of them is regarding the morbidity and mortality of COVID-19 vaccine, where we have talked about all 4 different platforms of the vaccine. We also have submitted the reinfection paper. That's a very fantastic paper where we have seen the timeline of when do you get reinfected versus vaccinated in comparison to non-vaccinated across different variants between Delta, Alpha, and Omicron. We also talked about the safety and efficacy of the homologous versus heterologous inactivated and mRNA vaccine that has been also published to the has been submitted to the vaccine journal. And finally, we have the overview of COVID-19 vaccine rollout in the Kingdom of Bahrain in collaboration with Pfizer company that has been submitted to Family Medicine and Community Health Journal. On this slide, among of the 31 publication, I have to say 20 of the publication came through the collaboration with the RCSI, with the students and with the different faculty members. So we have covered 5 aspects of epidemiology and public health. And I think one of the papers that Saad was talking about the CT value, and there was another paper with Dr. Kumar when we talked about the viral shedding and the transmission among in correlation with the CT values and the genotyping of the virus. We have done 2 publications in diagnostic that was about the antibody and the antigen test. And we had 11 publications. 2 of them are the RCT trials in clinical and treatment project, and finally 2 publications related to the vaccine. Next slide. To give you the overview of the all papers that we have published in collaboration with the RCSI faculty and students. I have to say, if you need a copy or a reference of those papers, Celine has a copy. We'll be more than delighted to send it to you. Again, quite extensive range of publication that covers all the different aspects of COVID-19. And as I said, all of this has been translated into the data-driven decision-making policy in the Kingdom of Bahrain. Next slide, where I'm going to present the first randomized controlled trial in the Kingdom of Bahrain was about the effectiveness and the safety profile of plasma. Now you have to remember, we have done this paper back in April 2020, where there has been really limited data, or actually no data that says whether plasma therapy has been effective. Remember, at that time, we have no therapeutic that has been standardized to be used for COVID-19. So we lack the data and we lack the therapeutic options. And rather than waiting for patient to die from SARS-CoV-2, we had to use the best available therapeutic option. And plasma therapy has been very well known in treating viral pneumonia. And the first papers has been launched was for H1N1, influenza, and also for MERS-CoV-1 5 years ago. So we wanted to see the effectiveness of plasma therapy at that time, and we have really used it in a randomized controlled trial, and we had some limitation, and I really always like to begin with the limitation. Recruiting the patient was not really easy to begin with. Again, was very difficult to convince either the patient or the family to be part of a clinical trial. So that really took us a lot of time to kind of set the scene and the theme for the Bahraini community to understand what's the clinical trial means. And of course, we have been working very hard with the NHRA and other institutions to recruit the patients. So what we have done basically, just a very brief background, is plasma therapy was effective in preventing the complication of SARS-CoV-2, and was it safe? Again, back then, there has been very limited data, but I'm also happy to say that we have also engaged with Stanford University, and we have engaged with NIH, and we have really had one of the largest systemic and meta-analysis research group, where there has been, I think, 32 countries involved, Bahrain among of those countries, where there has been 19 RCT with 32 non-RCT when they looked at the effectiveness of plasma, and that had published in the scientific report in February 2021. Our result which was really before the publication of this, has been really in keeping with many international publications when it comes for the effectiveness of the plasma therapy. So basically, that has been back in April till June 2020. We have enrolled a total of 40 patients. And again, the idea was to infuse them with 200 ml of convalescent plasma over 2 hours and compared to the placebo group. Next slide. So when it comes for the methodology, if you go to the next slide, there has been 2 major outcomes that we really wanted to study. Now, remember, again, back then, if you go to the next slide, back then, we didn't know that the dynamic of the viral illness has biphasic, which is the viremia and the inflammatory phase. So if you go to the next slide. So on the primary outcome that we have focused was basically do patients that receive plasma versus the standard of therapy, which is nothing, gonna be requiring to be on non-invasive ventilation or mechanical ventilation. And again, the whole idea is to prevent those patients from being intubated or requiring higher flow of oxygen. And the secondary outcome, again, we're tapping the second part of the viral illness, which is the inflammatory phase. Do they have high elevated C protein, procalcitonin, and lactate? And of course, all the other clotting factors like the D-dimer and the ferritin. Will the plasma therapy has an effect in reducing the effects of those markers? So of course, we have done extensive laboratory measurement, including the CRP, the D-dimers, and ferritin, and other biomarkers. On the next slide. So basically, on the next slide, we're going to talk about some inclusion and exclusion criteria. So we conducted the study between April 2020 till June 2020. Of course, we have only enrolled those who have informed consent, and I said that was one of the difficulties in explaining to our community what the trial means and what's the end of it. Most of the people get afraid of using something that's under trial. And we have tackled only adults, 21 and above. And of course, one of the criteria, patient has to be on oxygen. So if your saturation above 92 or PA2 less than 60, or if IA2 300 or less. So this is all the international definition of hypoxia, or if you have pneumonia by chest imaging. And of course, the reverse would be for the exclusion criteria. Next slide. So we have done it in a randomization fashion. So that was in the block of 4, and that was a computer generated. Either you receive a placebo versus the convalescent plasma. Now, I have to say, for the convalescent plasma, we measured the antibody level. So basically, the whole idea from it is you want to give the patient the neutralizing antibodies to fight the virus and to prevent from the virus from attaching to the cells and causing further damage to your organs. So you had to measure the neutralizing. Again, back in April 2020, there has been no commercial test available in the market to look at the quantity. So we have used a very basic qualitative test with a semi-quantitative, and that was actually a kit from Singapore. Bahrain was the first country in the region to receive that kit and test. So we have used that test. Again, there has been some limitation using that kit, but that was the only kit available to measure the semi-quantitative of the neutralizing antibody of the donors. So randomized basic you receive plasma versus no plasma, and then we have looked at their labs and the requirement or the clinical aspect requirement of non-invasive or mechanical ventilation. And also we have studied the adverse event, how many of those who receive the plasma would develop, for example, heart failure, clotting, stroke, or PE. That has been one of the adverse events, especially in elderly when it comes for plasma therapy. And that was based on previous literature on using plasma therapy on viral pneumonias. Next slide. Again, Dr. Manal was one of the leading person that she has been looking at the convalescent plasma donors. Again, that has been done through the BDF hospital. So the criteria for the donors, and that's the international standard for any donors. So informed consent. We avoided women during that has been pregnant, or during the childbearing, because as you know, pregnancy can produce some antibodies and that can have an effect. So that has been excluded. Again, they should have a documented negative PCR from the respiratory, and it was nasopharyngeal back then. They should be symptom-free. Patient should be at age not less than 21, and the weight not less than 50 kg. And again, that's all. And we have screened for other other infectious such as HIV, hepatitis, and syphilis. So that was part of the international donor screening. Next slide. So the bottom line, and again, when you go to the next slide, Naveen, again, back in June 2020, when we have said the plasma therapy did not show any significant in terms of the length of stay, in terms of requirement of non-invasive or mechanical ventilation. And in fact, that has been really most of the follow-up literature that supported that plasma therapy did not have a major impact on preventing the complication of SARS-CoV-2. However, we have proven that to be safe. We did not have any patient, even though it has been a very small sample size, but we did not record any patient that died from the plasma therapy, neither developed heart failure, neither acquired PE or any clotting disorder. And I think that's because of our strict eligibility criteria. So there has been no significant at all when it comes for the clinical outcome. Next slide. Things we have learned from this first RCT and the limitation, when you go to the next slide. Again, that has been a very small pilot study to introduce the safety of the plasma and maybe to study the efficacy of the plasma therapy in a larger scale. And as I said, there has been done several publications, like 19 RCTs and more than 32 non-RCT that all showed that there was no significant benefit. And that's why we have removed plasma therapy from, it used to be one of the recommendations in our protocol, but then we have left it only if you use it under clinical trial, you can use the plasma. Other than that, you should not give routinely to all patients. Limitation, again, as I say, sample size, and one of the biggest limitation was we did not have a fully quantitative machine that can detect the neutralizing antibodies. Most of the study that has been published in 2021, again, at that time we had the commercial kits available for quantifying the neutralizing antibody, because you should have a certain amount of neutralizing antibody to give to the patient with COVID-19. But even with a very high neutralizing antibody, that did not show any clinical significance. So to make it short, at the end, we have concluded that plasma therapy was at least safe, but was not effective in reducing the length of stay of hospitalization, neither will reduce the requirement for non-invasive or mechanical ventilation. And that's the end of my presentation. Thank you very much.
Professor Manaf, thank you very much indeed for your inspiring talk and showing not only the extent, but also the quality of the studies that were being undertaken, with this being an excellent example of that. Could I have the next slide, please? I'd now like to invite Mr. Abdullah Awadi, the Research Coordinator for the National Task Force Research Working Group, to present on the effect of ABO blood group and antibody class on the risk of COVID-19 infection and severity of clinical outcomes. Thank you very much indeed.
Greetings everyone, and good afternoon. I hope everyone is doing well today. My name is Abdullah Ismail Awadi, and I'm a Research Data Analyst and Research Coordinator in the National Task Force of Combating COVID-19. As you know, COVID-19 is a very serious and devastating pandemic, and we are all aware of the consequences of its spread. I've been working hand in hand with Professor Minar since the beginning of the pandemic, and I'm hoping to present today a previous project of ours. This project was done quite early on in the pandemic and published in 2021. The title of this project was The Effect of ABO Blood Group and Antibody Class on the Risk of COVID-19 Infection and the Severity of Outcome in of these infections. It was published in Scientific Reports, which is under the umbrella of Nature. Now for our introduction. We are going to talk about why we chose to do this research and the importance of it and the objectives of this research. At the time of the study, there was no known biological biomarker that can predict the chances of you getting infected or the severity of the outcome of the infection. Thus, a lot of research was done on many different risk factors and biological biomarkers. The first observation that was reported in 2020 reported that blood group A showed an increased risk for severity and infection and likelihood of infection, while blood group O showed a decreased risk of infection and severity. These findings were replicated in multiple studies. However, there was an article published later on by Latz et al. And that article itself was quite contradictory to what was found previously. This article reported that the blood group B had the increased likelihood of infection and increased severity of the outcome. Well, and this kind of created a gap in knowledge, because some people were saying the blood group A, some people were saying the blood group B. And that in itself was why we set our objective to study such thing. We decided to do the analysis on two main things, two things: the likelihood of infection and the severity. And that's what we can look at later on in our methods and results. Moving on to the method section, we will be talking about the study design and the participants that were included in the study. This study was a cross-section observation study. We investigated the association between blood group and risk of COVID-19 infection and severity of clinical outcomes. The association was analyzed by ABO groups, blood groups, and blood antibody class. To study the effect of blood type on the severity of clinical outcomes, the distribution were compared between with people infected with COVID-19 that were admitted to general COVID facilities and people that were infected with COVID-19 and are admitted to ICU COVID-19 facilities. This was replicated for both groups, for both analyses, where we looked at the blood groups and the antibody class, where anti-A was blood group B and O, and anti-B were blood groups A and O. The participants, we took a random sample of 3,000 COVID-19 positive individuals from the national database. Of those, only 2,138 individuals had blood group data documented or obtainable, thus were included in the study. As of July 2020, we only had 196 COVID-19 cases with ICU admissions, all of which that we went and we got their blood groups data and were included in the analysis. The blood group data for all COVID-19 infected individuals were obtained from medical records that were already available to us. Patients included in the blood type analysis were representative of the general distribution of blood groups in Bahrain. And to determine that, we went and looked at 2 years back into almost 5,000 samples donated to King Hamad University Hospital. And we had a similar distribution of blood groups within our analysis and the general population of Bahrain. Now to our results, where we will discuss the two analyses done, where one we handled the likelihood of infection and the other was where we handled the severity of the outcome. Now to the results. Chi-squared and Fisher-exact tests were used to compare the distribution of blood groups and antibodies between samples. Odd ratio tests were used to study the odds of blood type or antibody category tests to testing positive. In a 1 versus all manner, odd ratios were presented with 95% confidence interval. Our data does not present any association between the susceptibility to COVID-19 infection and blood group A similar to Latz et al. In addition, we found a significantly increased risk associated with blood group B, which is also comparable to both Latz et al. And a meta-analysis done by Liu et al. Individuals with blood group AB showed a decreased risk of COVID-19 infection, which is in line with the multiple reports. However, like other reports, we face the issue of a small sample size for the AB blood group. Unlike all other reports, no association between blood group O and decreased risk of infection was observed. Now, moving on to comparing the COVID-19 admission versus COVID-19 general admissions and COVID-19 ICU admissions. There was no association found between blood groups and susceptibility to more severe COVID-19 infection, although we have observed a decrease in odds of individuals with blood group O requiring ICU admissions, which is in line with Zhao et al. However, it wasn't statistically significant. Hence, no association was observed between the blood group or antibody class and the chances of getting severe COVID-19. And finally, for our conclusion, in an ideal world, our data would have been conclusive or even revolutionary. However, we do not live in an ideal world, and our data, even though it showed some statistically significant differences, it had its own fair share of limitations due to sample size for the AB blood group and such things. Results from the study and limited data available for us during the time of the conduction of the study makes the findings of our research quite hard to take for granted, or let's just put it in this way, quite hard to interpret and make a strong argument with. However, the contradiction that we observed in this field or in this part of the research regarding COVID-19 could be actually due to some factor that we did not look at, or some other reports did not look at, thus yielding different results every time the data the research method was replicated. Thus, such things could be looked at in the future project, or we could even look at it now that we have more of a database that is available to us due to the fact that the increase in ICU admissions and COVID-19 infections. Thank you all for listening, and I hope you have a great rest of your day, and we will be with you for the Q&A afterwards.
Thank you very much indeed, Dr. Abdullah. That is a fascinating study. And I was going to mention at the end, but I have to say, this has led to a huge amount of attention. And we've recently just been told, in fact today, that this was in the top 100 of all scientific reports, publications that have been downloaded. And in fact, it was number 61, which is an amazing achievement. And so well done, Professor Manafa and Abdullah. Brilliant. So finally, I would like to invite Dr. Nigel Stevenson, our Senior Lecturer in Immunology and Biochemistry at RCSI Bahrain, on his presentation, Investigating Viral Immunity, Implications for COVID-19. Thank you very much, and the floor is yours, Nigel.
My name is Nigel Stevenson, and I lead the Viral Immunology Research Team. I'd like to thank the organisers for inviting me to speak today and tell you about some of my lab's discoveries in relation to viral immunity and its implications for COVID-19. As we know, we live in a wonderful world. But we're continually posed with a major global threat. And of course, since we're currently living through a global pandemic, we know only too well that the threat to humanity is from viruses. And the threat from viruses has risen with an increase in newly emerging viruses such as SARS coronavirus 2 that causes COVID-19. There has also been an increase in animal to human transmission of viruses. Indeed, we have most recently seen how coronaviruses can jump from bats to civet cats or bats to camels, and then on to humans, with devastating effects. And of course, we've seen an exponential rise in global travel, which facilitates this spread of viral infection to every corner of the world. Indeed, over the past 30 years, we've seen major viral outbreaks of HIV, Hepatitis C, SARS coronavirus 1, MERS coronavirus, Ebola, Zika, and of course, most recently, SARS coronavirus 2. However, with the exception of Hepatitis C virus, we still have no therapeutic cures for any of these other viruses. Therefore, as medical scientists, we are posed with the question, how do we fight and cure viruses? And of course, as an immunologist, I believe the solution lies with our immune system. So over the past 20 years, my research has been investigating the interplay between viruses and the immune system, with the overall aim of revealing novel targets for therapeutic intervention and cure. And my research group has specifically focused on the immune evasion strategies of 3 viruses: the Hepatitis C virus, respiratory syncytial virus, and human immunodeficiency virus, which all subvert the antiviral immune responses, leading to an increase in infection and the diseases that we see here. So during infection, pathogen recognition receptors, which are on the surface and within our cells, detect viral components such as viral RNA, DNA, and viral proteins. These pathogen recognition receptors activate intracellular signaling that leads to the production of both pro-inflammatory and antiviral cytokines, including type 1 interferons such as interferon alpha. Interferon alpha signals through an intracellular cascade of proteins called the JAK-STAT pathway. Once interferon alpha binds to its cell receptor, intracellular tyrosine kinases called Janus kinases, or JAKs, become activated. These JAKs then phosphorylate tyrosine residues of the receptor, providing docking sites for transcription factors called signal transducer and activator of transcription, or STATs. STAT proteins bind the receptor chains and then become activated. Once activated, STATs dissociate from the receptor and form dimers, which translocate to the nucleus and bind DNA promoter regions, leading to the upregulation of hundreds of antiviral genes called interferon-stimulated genes, or ISGs. These ISGs have a broad range of antiviral mechanisms. For example, MXA can physically bind and sequester viral proteins. OAS promotes cleavage of viral RNA, and PKR activation reduces viral RNA translation. And it's this broad range of antiviral effects that promotes viral clearance. However, we know that viruses are not all cured naturally, because they were brilliant at evading our immune responses, and in doing so, they cause disease and even death. So my lab's main research question has been, how do viruses evade innate immune responses? And since type 1 interferons are key to curing viral infection, we've been specifically investigating if viruses target the JAK-STAT pathway, thereby inhibiting antiviral responses to interferon alpha. And to investigate this, we turn to the homeostatic process of protein recycling that continually occurs within our cells. Suppressor of cytokine signaling, or SOX proteins, that contain a specific motif called a SOX box, are able to bind alongin C and cullen proteins to form an active E3 ligase enzyme complex. And this promotes the ubiquitination of target proteins. And these ubiquitinated target proteins are subsequently degraded in the proteasome, thereby releasing peptides and amino acids for further protein production. Our bioinformatic analysis has discovered that several viruses contain SOX box motifs within their genomes. And our studies have found that RSV, HCV, and HIV all use their SOX box containing proteins to form an E3 ligase complex, thus hijacking this process to promote ubiquitination and proteasomal degradation of STAT proteins. Indeed, we have found that without STAT proteins, normal responses to type 1 interferons are hindered, making the cellular environment optimal for viral infection and replication. So the first virus that we found to contain a SOX box was RSV. RSV is a seasonal winter infection. It mainly affects children and the elderly. It is estimated that RSV causes 64 million infections and 160,000 deaths each year. RSV causes a lower respiratory tract infection that can progress to bronchiolitis and pneumonia. Even though RSV causes so much disease, there still remains no vaccine nor effective therapy. And the standard treatment of care for RSV has been humidified oxygen and fluids. So having discovered a SOX box within the gene that encodes for RSV's non-structural 1 protein, we wondered if RSV could block responses to interferon alpha by promoting the proteasomal degradation of STAT proteins. Our first study, carried out when I was in Queen's University of Belfast, found that the NS1 protein of RSV promotes proteasomal degradation of STAT2. And here you can see when we express the viral protein, STAT2 is degraded, but this does not occur when treated with proteasomal inhibitors. Currently, Claudia, a PhD student in my lab, is analyzing the effect of RSV in epithelial cells, which are the main target for RSV infection. Claudia has found that when she expresses RSV's NS1 protein in either Bs2b or A549 epithelial cells, Several ISGs, such as MXA and PKR, are significantly reduced, highlighting the effect of STAT degradation on antiviral gene expression. Our second study focused on the immune evasion mechanisms of the hepatitis C virus. 180 million people are estimated to be infected with HCV worldwide. HCV readily infects hepatocytes of the liver, but has also been shown to infect immune cells. HCV infection often remains asymptomatic for many years, with patients only being diagnosed after symptoms of liver fibrosis develop. This means HCV infection can often silently progress to liver fibrosis and cirrhosis, which can further develop into hepatocellular carcinoma, and in some cases, liver transplantation is required. For many years, HCV had been treated with therapeutic interferon alpha plus ribavirin, but over the past 2 decades, new direct-acting antivirals have been developed that provide a cure for a large proportion of HCV-infected patients. However, with interferon alpha's diverse antiviral response, we would expect a high level of viral clearance. But up to 85% of HCV-infected patients do not clear the virus naturally, and up to 50% of genotype 1 infected patients did not respond to interferon alpha treatment for reasons that remained poorly understood. So, as with RSV, we wondered if HCV could block responses to interferon alpha by promoting the proteasomal degradation of STAT proteins. Indeed, we discovered significantly lower levels of STAT1 and STAT3 protein in both peripheral blood mononuclear cells and liver hepatocytes from HCV-positive patients, which may explain the poor response to therapeutic interferon alpha. Having observed a loss of STAT proteins in cells from HCV-infected patients, we wondered if this degradation could be blocked by inhibiting the proteasome with an inhibitor called MG132. We found that proteasomal inhibitors restored STAT protein expression, as you can see here with STAT3, and ISG induction, as you can see here with MXA, indicating that, like RSV, HCV also promotes proteasomal degradation of the interferon JAK-STAT pathway, which in turn suppresses antiviral gene induction. So having seen that RSV and HCV block responses to interferon alpha via STAT degradation, we next investigated the effects of HIV upon interferon signaling. It's estimated that 38 million people live with HIV infection worldwide. But thankfully, antiretroviral therapy now largely avoids progression to AIDS, thus reducing HIV-related mortality. However, these therapies often have side effects and are required to be taken for life, making them extremely expensive. Interestingly, in the 1980s, interferon alpha therapy was trialed as a therapeutic for HIV, but its poor response did not cure the infection, suggesting to us that the virus may be blocking the interferon pathway. Furthermore, while up to 50% of mono-infected HCV patients responded to therapeutic interferon alpha, this response rate dropped to only 30% in HCV/HIV co-infected patients, further indicating that HIV blocks the interferon pathway. Indeed, it was these clinical facts and the presence of a SOX box within HIV that led us to investigate if, like RSV and HCV, HIV could also block responses to interferon alpha by promoting proteasomal degradation of STATs. Dr. Kieran Bannon, a medical PhD student in my lab, found that both STAT1 and STAT3 protein levels were reduced in PBMCs from HIV-positive patients compared to healthy controls. And Kieran found that when we treated these cells with interferon alpha, STAT phosphorylation, and antiviral ISG induction were significantly reduced. So having observed a loss of STAT proteins in cells from HIV-infected patients, we wondered if this degradation could be blocked by inhibiting the proteasome with MG132. Siobhan Gargan, a postdoc in my lab, found that HIV VIF protein, which contains a SOX box, promoted the proteasomal degradation of STAT1, and that this degradation could indeed be blocked by inhibiting the proteasome with MG132. Yamei Zhang and Aziz Alataibi, PhD students in my lab, are continuing these studies by analyzing the immune evasion mechanisms of the coronaviruses MERS, SARS-1, and SARS-2. Indeed, Yamei already has preliminary results to show that expression of MERS or SARS-1 proteins significantly block the induction of antiviral ISGs, as can be seen here with MXA and ISG15, indicating that these coronaviruses also suppress the interferon pathway. So to summarize, we found that RSV, HCV, and HIV all promote proteasomal degradation of STAT proteins, which in turn suppresses the optimal induction of antiviral ISGs, effectively allowing these viruses to avoid elimination. I'd like to finish off by acknowledging the hard work, determination, and enthusiasm of my research team, and all the kindness of our collaborators, and of course the funding bodies who've made all this research possible.
Thank you very much indeed, Nigel. That was very thought-provoking, and I'm sure it's going to lead to some very interesting questions in the question and answer session. So I'd now like to undertake the question and answer session, and looking at the questions that have come in, I'll take them in kind of order, as it were. The first question is to you, Saad. So the question is, so do you think that comorbidity kind of trumps viral load? You seem to show that viral load didn't equate to severity. So do you think it's the comorbidity of the patient which is more important?
Thank you for that, Professor Apkin. Definitely, there has been a lot of studies that have already confirmed a definite association between a lot of comorbidities and COVID-19 disease severity versus our study and several others which show that viral load is more debatable in that sense. But age, for example, has been shown to be a definite comorbidity, with the elderly being most at risk, and a lot of other medical conditions as well, which our study has also showed. And we have other publications and other papers one of them which was specifically on a couple 100,000 Bahraini patients, and we saw that a lot of comorbidities related specifically cardiovascular disease and obesity were definitely associated with severe disease.
Thank you. Professor Manaf, the plasma study was generally kind of a negative study, but do you think that was a universal outcome for all variants? Do you think that different variants have a different response?
Right. So our paper looked at the original virus, so the Wuhan virus, and most of other people that came in 2021, they actually have looked at Delta, they have looked at the Alpha variant, and all of them had the same end results. So plasma therapy was not effective, and I think that's why it has been removed from the recommendation from the NIH, from the CDC, and from the WHO.
Okay, and I know that unfortunately Abdullah can't be with us, but Professor Manaf, you very kindly agreed to answer his questions as well. So you're going to double whammy, really.
No problem.
So obviously what Abdullah did highlight at the end, there was a number of limitations to the study, which, I mean, one of the issues was an ethnic difference possible? Do you think that ABO, blood group and ethnicity could be a factor?
So that was the theory. So let me go back and just explain the rationale behind that paper. So back then in the earlies of the pandemic, there has been lots of reports that came across that if you have blood group A, you tend to have more at risk of acquiring the virus and might be at risk of having a complication such as death or being admitted to the ICU. So we really have studies around 2000 proven to be COVID, and we compare it to the controls, which is non-COVID, when we went back and reviewed all the national database of blood group type in the Kingdom of Bahrain. And again, not surprisingly, we did not see any effect of being whether blood group A or B put you at risk of acquiring the virus, and neither that put you at risk of admitting to the ICU. The other interesting part is actually Bahrain. The majority of the Bahraini population is blood group O, and again that was reflecting in the papers. And most of the European, which again most of the report came across, was between blood group A or AB. And I think there has been many other co-founding, being whether AB or group A late presentation. And I think that was one of the highlight of SARS-CoV-2 late presentation that tend to be worse in outcome. And I'm just reading for other recent papers that tackle the blood group, and again did not find any significant whatsoever in terms of the type or even the class of the blood.
Thank you very much indeed. That was your very comprehensive answer. Saad, turning to you. Obviously, this work was, it's mentioned, done on the original, as it were, SARS-CoV-2 virus. Do you think that if you had a more virulent virus, that it would change the viral load, that would give you the CT values that may then reflect in symptoms?
So I think that's a good question, but it's worth remembering that we also had a large proportion of patients with a CT value between 10 and 20, and that's very, very low. So that would be a very high viral load. So I don't think that if we had a larger proportion of people with higher viral loads, we'd necessarily find a correlation. And even if we did, again, what's the clinical correlation there? That would answer the biologic question. But there's a lot of study designs that are more appropriate for that biological question. I think in Professor Nigel's lab would be more a better place to find an answer for that. But clinically, I don't think there would be much of a difference. But if when we played around with the analysis as well, and we tried to we excluded one of the confounding groups, which was the migrant workers. So we focused on Bahrainis. The data started changing a bit. So I do think there's room for more studies focused on a more homogeneous cohort versus ours, like I said, which was very heterogeneous.
Thank you very much indeed. Again, Professor Manaf, the plasma study again was, as I say, negative, but do you think that was due to the fact that we just needed a bigger cohort? And were we looking at the right markers? I mean, one of the concerns is that we just weren't looking at the right markers and correlating those to the outcome.
Sure. So before I answer your question, I'm going to go back to Saad point, as he clearly mentioned. Again, we have looked into this, the correlation between the variance and the CT values in another study with Dr. Kumar. And in fact, in theory, you'd expect, for example, when it comes for the alpha, delta, and even now the Omicron, is that because of the transmission is so high, so they should have higher viral load, and therefore they're going to transmit the virus. Now, one caveat is CT value does not really reflect to the actual viral load. It just gives you a fraction of the genome, so you cannot correlate clearly if the CT value at this level means you have either high or low viral load. But in fact, with the Delta, we had patients with higher CT value, meaning they have a low viral threshold or viral load, and they are more transmittable than in comparison, for example, to the Alpha. Omicron, completely the opposite. Now you're talking about for the R naught for Omicron is actually up to 9. So close to measles, in fact. And again, we had people when they present initially, and they have a high CT value, meaning they have low viral load, if you want to say viral load. So there was really no correlation between the CT value and the viral transmission. And in Nitya paper, she actually documented it was not statistically significant between the CT value and the viral shedding. So that just to kind of close that part. And it's quite fascinating if people are interested to look at Dr. Nitya Kumar paper. Back to your question, definitely size matters. And that again has been one of the challenges. And yes, when it comes for the inflammatory markers, we did not look into the interleukin 6, which is again going to be a key marker in terms of the progression of the disease. But again, that was early of the pandemic. No one really looked at interleukin 6, and we did not have it back then here, even in the Bahrain. But right after this papers, we had the interleukin 6, we had the interleukin 1 and 2 and 3. So all of this are additional vital markers that we did not look into during the plasma therapy paper.
So turning to you again, Professor Manaf, to answer the question on behalf of Dr. Abdullah. In the presentation, there was quite a few patients that had to be excluded because you just didn't have their blood type. Yeah. And one of the limitations that you mentioned, despite the fact you had a large number of patients, do you think that the patients that you couldn't actually measure introduced bias into the results that might have actually prevented you from actually getting a positive result?
Absolutely. And again, one of the key limitations, and this is something that we try to overcome in Bahrain when it comes for the data collection, is that you don't have a centralized data that's available. So we really worked very hard to go across between the Ministry of Health to the King Hamad and even to the BDF to get the blood type. After this paper, all of this information has been centralized. So every COVID, for example, at the moment, you will get automatically their blood group. If you don't have it in the system, the nurse gonna take and collect it. So as you said, we really learned a lot from all of this research, and we really had to fine-tune our database here in the Kingdom of Bahrain. And I'm very glad to say, for the first time, when we have all 3 major hospitals in the Kingdom of Bahrain now, they're interlinked only for COVID. For now, where you have all the information that's available from the demographic, from the comorbidities, and from the blood type group as well.
So Nigel, I'm afraid you're going to get 2 in succession. But okay. Just come in. So does this mean our natural type 1 interferon doesn't work effectively or efficiently to clear infection during the COVID-19?
Yeah, we believe that that's true, Steve. So if you are removing the components of the pathway that interferon signals through, you can't upregulate all the antiviral genes efficiently. So it's one of the observations that a lot of people have seen is that interferons are actually increased or in normal levels in COVID patients. And we have this cytokine storm as well, where you have lots of cytokines, but it's not necessarily getting rid of the virus. So we believe that it's actually the pathway is being destroyed, and it may be even beyond interferon. Manaf mentioned interleukin 6. Interleukin 6 uses STAT3 to induce its pro-inflammatory effects. And if the JAK-STAT pathway has been targeted, it may not be just specifically to remove response to antiviral interferons. It may be other pro-inflammatory as well.
And secondly, are there any other viruses that might use this mechanism to kind of block responses to interferon?
Yeah, so I think probably my career is panned out for the next 20 years because we have identified with our bioinformatic analysis that other viruses have the potential to do this, including dengue virus, Ebola as well. So some of the major viruses, and we're seeing these conserved mechanisms and these conserved regions within their genomes that indicate that viruses have actually evolved to use the mechanism that we're investigating. So in answer to your question, yeah, I don't think it's stopping at HIV, RSV, hepatitis C and coronaviruses. And potentially, if this has been a mechanism in a region, it could be in the next emerging virus as well. That's something we'll be watching out for.
And what you showed also very interestingly in your actual presentation was ubiquination of the proteins that then led through its destruction in the proteasome. And of course, one of the classic mechanisms by which this occurs is kind of upregulating or altering the heat shock proteins. Are the heat shock proteins intimately involved in many of these kind of areas?
It's absolutely possible. It's a really good point because the heat shock proteins, of course, are linked to IL-6 as well, and they're linked to the liver. So whether or not they are ubiquinated by the viruses, we haven't looked at that, but that would be a really nice thing to see if ubiquination promotes their degradation, or even sometimes ubiquination can cause stabilization in certain circumstances, and you could have more inflammatory effects from the heat shock proteins. Yeah, so it could all be interlinked.
So Saad, final question for you. And that one of the things that I think you showed on the second or third slide was about symptoms versus no symptoms. So you were talking about the patients who got symptoms with mild, moderate and severe disease and their CT values. Was there a cutoff for those patients who did not get any symptoms at all?
Sorry, do you mean a CT value cutoff specifically?
CT value cutoff. So you know that they're infected. Were they universally a kind of a high CT value, or was it again a spread?
So a CT value more than 40 was the one that we used, where if a patient had a CT value more than 40, we didn't even diagnose with COVID. But I think what you mean is specifically if symptomatic versus asymptomatic had different CT numbers, and the answer to that would be no, like there wasn't a significant difference in CT value between both groups. So it wasn't something that whether that means that CT value itself is not correlated with symptomatology, or whether that means that viral load is also not correlated, we can't tell just from that, but that would be interesting as well.
And Professor Manaf, what lessons did you learn from the plasma trial, undertaking the plasma trial?
Well, it's two things. The first is that neutralizing antibody would be too late in the inflammatory phase. So you should really use it before patient requiring an oxygen. I think this is something might be some useful. And in fact, sotrovimab, which is one of the synthetic monoclonal antibodies, has been labeled to be used before patient requiring to be oxygen. Two, you should have a certain level of neutralizing where you should say it's efficient. And I don't think there's an agreed standard which level. But however, the synthetic one, which is the monoclonal antibodies, they have proven to be effective in preventing the complication. And in fact, we have looked at sotrovimab in a very small pilot, around 108 patients, been admitted to the ICU, and they are on oxygen, but they're less than 6 liters of oxygen. Again, this is just very preliminary, and 78% of those who received sotrovimab, the average age was around 78, mainly Bahraini, with multiple comorbidities. 78% actually has been discharged home from the ICU off oxygen. We had 8 patients out of 108 died, but it was not COVID-related death, was something else. So this is just to kind of summarize. So the plasma will not be very effective if you use it during the inflammatory phase. If you want to try the monoclonal, then I think that's something will be interesting, and we're waiting for the data to prove it.
Thank you very much. So that's the end of all the questions that have actually come through. I would like to thank all the presenters for their first-class presentations. And again, I would like to emphasize the quality of the presentations, particularly the ABO antibody, the ABO blood group study, which was so highly cited in scientific reports. So many thanks for your attendance and all your questions. And I'd just like to draw the meeting to a close, and I hope everyone has a good evening. Thank you very much indeed.